Hello. Welcome back to Following Science, the podcast where we explore the latest and most exciting discoveries in science and technology. I'm your host, Ruto Parle, and today we are diving deep into the fascinating world of single cell metabolomics. In this episode, we're going to cover everything from the basics to the cutting edge technologies enabling single cell analysis and explore real world applications that are transforming our journey and understanding of biology and medicine. Now, make sure to grab your coffee. I have mine. And let's embark on this journey together. Now, to truly appreciate single cell metabolomics, we need to start with the basics. What is it? Well, metabolomics is a comprehensive study of small molecules, commonly known as metabolites, within cells, biofluids, tissues, or organisms. These metabolites and their interactions form metabolic pathways, which are essential for maintaining the cellular processes that sustain life. Just like coffee is essential to keeping us all awake. Now, you're gonna hear me bring up coffee a lot this episode just because I drink so much of it, but please bear with me. Now, let's dive right into the history. Metabolomics emerged as a field in the early 2000s, building on the foundations laid by genomics and proteomics. Now, I already explained that the term metabolome refers to the complete set of metabolites in a biological cell, tissue, organ, or organism. But what I didn't mention is that this is all the end products of cellular processes. Now, while genomics and proteomics focus on genes and proteins respectively, metabolomics offers a direct reflection of the biochemical activity within a cell. Now, metabolomics can be divided into two main branches, targeted and untargeted. Let's start with targeted. Now, targeted metabolomics focuses on quantifying a specific set of known metabolites, whereas untargeted metabolomics aims to measure as many metabolites as possible in a given sample without prior knowledge of their identity. Now, both approaches obviously have their own advantages and applications like everything in life, but today we're going to focus on specifically untargeted metabolomics, as it aligns closely with the goals of single-cell analysis. Now, understanding metabolism is also crucial for grasping the significance of metabolomics. Now, what we all know about metabolism, or as I'm guessing, is the fact that it keeps you thin, because high metabolism means you process things quicker, which means that you'll stay thin. But there's a lot more to it. For example, metabolism encompasses all the chemical reactions that occur within a cell to maintain life. These reactions are organized into metabolic pathways such as glycolysis, the citric acid cycle, and the electron transport chain. Now, any of my fellow listeners who are AP Bio fans or just have studied it before know what I'm talking about by these metabolic pathways. And if you don't, that's okay, because we'll cover it in a little more detail later on in this episode, and or I can create a new episode just for y'all. Now, each pathway in this metabolic pathway plays a specific role in converting nutrients into energy, building cellular structures, and lastly, eliminating waste products. Now, the study of metabolites provides a snapshot of the physiological state of a cell or organism, making metabolomics a powerful tool in various fields, including drug development, disease diagnosis, and personalized medicine. By analyzing the metabolome, researchers can gain insights into disease mechanisms, identify potential biomarkers, and evaluate the effects of therapeutic interventions. Historically, metabolomics has been applied to numerous biological and medical research areas. For example, in cancer research, metabolomics has helped identify unique metabolic signatures associated with different cancer types, paving the way for the development of targeted therapies. Now, in environmental science, metabolomics has been used to study the effects of pollutants on ecosystems by analyzing the metabolic changes in exposed organisms, which honestly sounds super cool. Now that we have a solid understanding of what metabolomics really is, let's delve into single-cell metabolomics. Traditional metabolomics typically analyzes bulk samples, which provides an average metabolic profile of a population of cells. Now, while this approach is powerful, it overlooks the cellular heterogeneity within tissues. Single-cell metabolomics, on the other hand, allows us to analyze the metabolites of individual cells, providing unprecedented insights into cellular diversity. So, what exactly is single-cell metabolomics? At its core, single-cell metabolomics involves the extraction, identification, and quantification of metabolites from individual cells. This approach is essential because cells with the same tissue can exhibit vastly different metabolic states, which can have significant implications for understanding biological processes and disease mechanisms. For instance, in cancer, tumor cells can display diverse metabolic phenotypes that contribute to disease progression and resistance to treatment. By studying individual cells, researchers can identify unique metabolic pathways that drive these processes, leading to the development of the more effective therapies. Now, the impact of single-cell metabolomics on biological research cannot be overstated. It has opened new avenues for investigating cellular functions, disease mechanisms, and therapeutic targets. By revealing the metabolic intricacies of single cells, this approach is transforming our understanding of biology and medicine. Moreover, single-cell metabolomics is crucial for studying rare cell populations, such as stem cells and circulating tumor cells, which may be masked by the dominant cell types in bulk analysis. This level of detail allows researchers to explore the roles of these rare cells in health and disease, providing insights that were previously just unattainable. Now, the advancements in single-cell metabolomics has been driven by cutting-edge technologies. One of the key technologies enabling this field is mass spectrometry, or MS, or just mass spec. MS is now a powerful analytic technique that measures the mass-to-charge ratio of ions, allowing for the identification and quantification of metabolites. Let's dive into how this technology works and the applications of it in single-cell metabolomics specifically. Now, in single-cell metabolomics, MS is often coupled with advanced imaging techniques to precisely target and analyze individual cells. Techniques such as matrix-assisted laser disruption ionization, or MALDI, and secondary ion mass spectrometry, or SIMS, or SIMS, are commonly used. These techniques enable the high-resolution analysis of metabolites in single cells, providing detailed metabolic profiles. Now, MALDI or MS involves embedding the sample in a matrix that absorbs laser energy and assists in the ionization of analytes. When the laser hits the matrix, it causes the analytes to desorb or ionize, allowing them to be detected by the mass spectrometer. This technique is particularly useful for spatially resolved metabolomics, where the distribution of metabolites within tissue sections can be mapped. SIMS, on the other hand, uses a focused primary ion beam to bombard the sample surface, causing the ejection of secondary ions that are then detected by the mass spec. This technique offers high spatial resolution, making it ideal for analyzing the final metabolic details of individual cells. Now, another important technology is nuclear magnetic resonance, or NMR. NMR spectroscopy provides detailed information about the structure and dynamics of metabolites. Although less sensitive than MS, NMR offers unique insights into metabolic pathways and interactions within single cells. Now, NMR works by placing samples in a strong magnetic field and applying radio frequency pulses, which can then cause the nuclei of certain atoms to resonate. The resulting signals are then used to determine the chemical structure of metabolites. Now, despite its lower sensitivity, NMR has several advantages, including the ability to analyze complex mixtures without the need for extensive sample preparation and the capability to provide quantitative information. In single-cell metabolomics, NMR is often used in combination with other techniques to provide a more comprehensive view of cellular metabolism. Now, the field of single-cell metabolomics also faces several challenges. This includes the sensitivity required to detect metabolites in tiny volumes, the complexity of data analysis, and the need for robust and reproducible methods. Now, researchers are continually developing innovative solutions to address these challenges, such as enhancing the sensitivity of analytical techniques and improving computational tools for data analysis. For example, advancements in sample preparation techniques such as microfluidics have improved the ability to isolate and analyze single cells. Microfluidic devices allow for the precise manipulation of small volumes of fluids, enabling efficient capture and processing of individual cells. These devices can be integrated with MS or NMR systems to streamline the workflow and enhance the sensitivity of metabolite detection. Now, data analysis is another critical aspect of single-cell metabolomics. The first step involves the collection and pre-processing of data. Now, ensuring that high-quality data is essential for accurate analysis and interpretation. This involves meticulous sample preparation, calibration of analytical instruments, and rigorous quality control measures. Once the data is collected, various analytical techniques are employed to interpret the metabolic profiles. Statistical methods such as the principal component analysis or PCA and hierarchical clustering are commonly used to identify patterns and relationships within the data. These techniques can help reduce the complexity of the data and highlight key metabolic differences between individual cells. Now, computational models and machine learning algorithms are also increasingly utilized to extract meaningful insights from complex datasets. Machine learning can identify subtle patterns and correlations that may be missed by traditional statistical methods. These models can also be trained to predict cellular states or responses based on metabolic profiles, providing valuable tools for hypothesis generation and testing. Now, visualization of single-cell metabolomics data is crucial for understanding and communicating findings. Techniques such as heat maps, scatter plots, and metabolic pathway maps are used to represent the data visually. Heat maps, for instance, can display the abundance of metabolites across different cells, highlighting metabolic heterogeneity. Now, scatter plots can be used to show the relationship between different metabolites or to compare metabolic profiles across cell populations. Now, integrating metabolomics data with other omics data, such as genomics and transcriptomics, provides a comprehensive view of cellular functions and interactions. Multi-omics integration allows researchers to correlate metabolic changes with genetic and transcriptional alterations, offering deeper insights into the regulatory and mechanisms that drive and underlie cellular processes. For example, combining single-cell RNA sequencing with metabolomics can reveal how gene expression changes influence metabolic pathways in individual cells. This integrative approach can uncover novel regulatory networks and identify key drivers of cellular behavior. Now, let's explore some real-world applications of single-cell metabolomics through detailed case studies. One prominent example and application is cancer research. Single-cell metabolomics has provided valuable insights into the metabolomic heterogeneity of tumor cells. By analyzing the metabolites of individual cancer cells, researchers have identified unique metabolic pathways that drive tumor growth and resistance to therapy. For instance, in a recent study, researchers used single-cell metabolomics to identify metabolic profiles of different subpopulations of cancer cells within a tumor. They discovered that some subpopulations were highly glycotic, relying on glucose metabolism for energy production, while others were just more oxidative, depending on mitochondrial respiration. This metabolic diversity contributes to the tumor's ability to adapt to different environmental conditions and resist treatment. Now, by targeting these distinct metabolic pathways, researchers are developing new therapeutic techniques and strategies to overcome drug resistance and improve treatment outcomes. For example, combining glycolysis inhibitors with drugs that target oxidative metabolism can potentially eliminate both glycotic and oxidative cancer cell subpopulations, reducing the likelihood of treatment failure. Now, moving on to neuroscience, single-cell metabolomics is being used to study brain metabolism and neurodegenerative diseases. Researchers are uncovering how individual neurons and glial cells metabolize nutrients and respond to stress. These insights are then crucial for understanding diseases such as Alzheimer's and Parkinson's, where metabolic dysfunction plays a key role. One study investigated the metabolic profiles of individual neurons in the brains of Alzheimer's patients. The researchers found that neurons with higher levels of oxidative stress exhibited altered metabolism, with increased levels of certain metabolites involved in energy production and antioxidant defense. These findings suggest that targeting metabolic pathways associated with oxidative stress could be a promising therapeutic approach for Alzheimer's disease. Now, beyond cancer and neuroscience, single-cell metabolomics is also making significant contributions to immunology, metabolic diseases, and personalized medicine. In immunology, single-cell metabolomics is being used to study the metabolic reprogramming of immune cells during infection and inflammation. By analyzing the metabolites of individual immune cells, researchers can identify metabolic pathways that can support immune cell function and survival. For example, a study on T cells, a type of immune cell, revealed that activated T cells undergo significant metabolic programming, or should I say reprogramming, to support their rapid proliferation and effector functions. By targeting these metabolic pathways, researchers are developing new immunotherapies to enhance the effectiveness of cancer treatments and control autoimmune diseases. Now, in metabolic diseases, single-cell metabolomics is helping to unravel the complex metabolic networks underlying conditions such as diabetes and obesity. By studying the metabolic profiles of individual cells in metabolic tissues, researchers can also identify key regulatory nodes and pathways that contribute to disease development and progression, which should just help us move towards a future with more personalized medicine and more medicine focused on targeting like these single cell based on the single cell analysis, which is honestly just pretty cool. Now, let's move on to the future of single cell metabolomics before wrapping all of this up. Now, the future, as you probably heard before, is incredibly promising. As technologies continue to advance, we can expect even greater sensitivity, resolution, and throughput single-cell analysis. This will just enable researchers to explore previously inaccessible aspects of cellular metabolism and uncover new biological insights. One exciting prospect is the integration of single-cell metabolomics with other single-cell omics technologies such as genomics, transcriptomics, and proteomics. This multi-omics approach will provide a more comprehensive understanding of cellular functions and interactions, driving new discoveries and innovations in biology and medicine. For example, combining single-cell metabolomics with single-cell RNA sequencing can reveal how gene expression changes influence metabolic pathways in individual cells. This integrative approach can then uncover novel regulatory networks and identify key drivers of cellular behavior. However, with these advancements come important ethical considerations. Single-cell research involves the collection and analysis of data at an unprecedented level of detail, raising concerns about privacy and data security. It is essential to establish ethical guidelines and regulations to ensure that research is conducted responsibly and that the privacy of individuals is protected. For instance, when collecting single-cell data from patient samples, researchers must obtain informed consent and ensure that the data is de-identified to protect patient privacy. Additionally, data security measures must be in place to prevent unauthorized access to sensitive information. Now, looking ahead, the integration of artificial intelligence and machine learning with single-cell metabolomics holds great potential for accelerating discoveries and improving data analysis. AI algorithms can analyze large and complex datasets more efficiently than traditional methods, uncovering patterns and correlations that may be missed by human analysts. Now, moreover, AI can also be used to predict cellular responses to different treatments based on their metabolic profiles, guiding the development of personalized therapies. This approach has the potential to revolutionize precision in medicine, making treatments more effective and reducing the risk of adverse effects. Now we're done. Phew. We've covered a lot of ground in today's episode, so I hope you'll actually learn something from it. Single-cell metabolomics is transforming our understanding of biology and medicine, and I hope I just provided you some insights into how it is actually doing that. Now, as I wrap up this episode, I just want to thank you all for joining me on the scientific journey. Now, don't forget to rate, review, and subscribe to Following Science for more episodes exploring the latest in science and technology. Stay curious, and I'll see you next time. This is your host, Rachel Parle, signing off.